Good morning. It's April 29th, and this is your daily brief in science. Here's everything you need to know. A detailed map of about 1,100 olfactory receptors in the mouse nose shows they sit in tightly regulated spatial locations that align with smell maps in the brain's olfactory bulb. Retinoic acid acts as a key driver, with gradients guiding neurons to express the correct receptor type and changing the map if retinoic acid levels shift. This challenges the idea that olfactory receptor expression is largely random and lays groundwork for therapies to restore smell, including stem cell approaches or brain-computer interfaces. Led by Sandeep Datta at Harvard Medical School, with collaboration from Catherine Duloc's lab, the findings were published in Cell on April 28, 2026. The study was supported by NIH, the Youngstown Collective at Harvard, and NSF graduate fellowships. A separate study in the same Cell issue corroborates that a continuous transcriptional code and retinoic acid signaling established the dorsoventral organization of odor receptors. Datta and colleagues emphasize the broader significance of smell for daily life and mental health, noting that restoring olfactory function could improve safety, taste, psychological well-being, and advance basic sensory science. The work is described as a landmark that fundamentally changes understanding of the olfactory system and resolves a major question about how odor mapping occurs. The findings reflect a close collaboration across institutions and connect nasal receptor positioning to brain odor maps. Future directions include studying receptor stripes in human tissue to assess cross-species consistency and exploring implications for treating loss of smell and related risks like depression, with an eye toward translation to humans. UTMD Anderson unveils the Center for Cellular Language Intelligence, funded by a $10 million gift from Peggy and Carl Sewell. The center aims to decode the cellular language of cancer using spatial technologies, AI, and clinical insights to support earlier detection, smarter prevention, and more personalized treatments. It will map cellular organization, identify signaling hubs and programs driving initiation and progression, and translate high-dimensional discoveries into clinical targets and predictive biomarkers. This effort sits within a broader push toward spatial biology, functional genomics, big data, and AI to study cancer at the single-cell level and to understand tumor ecosystems and therapy response. The Sewells have a long history with UTMD Anderson, including roles on the Board of Visitors and funding the Sewell Family Distinguished University Chair, with Peggy named a life member of the Board of Visitors in 2024. Their philanthropy reflects a sustained commitment to advancing cancer research and patient care through board leadership and donor-led initiatives, with the endowment funding recruitment, infrastructure, and AI capabilities to manage large spatial datasets and foster cross-institutional collaboration. The gift is described as building an integrated hub for discovery, translation, and collaboration across disciplines to accelerate identification of therapeutic targets, biomarkers, and prevention strategies. Linghua Wang, an expert in single-cell and spatial biology, will lead the center, forming a multidisciplinary consortium blending genomic medicine, immunology, and data science to translate discoveries into clinical trials and precision medicine. The initiative will enable spatially resolved functional genomics to observe cellular responses in real time, guiding combination therapies and strategies to prevent resistance and relapse. Key methods include high-resolution single-cell sequencing, multiplexed imaging, and AI-driven analysis to map signaling networks, immune evasion mechanisms, and predictive biomarkers for immunotherapy. The project aligns with UTMD Anderson's fundraising campaign, Only Possible Here, which has raised over $2 billion toward breakthrough innovations and patient care improvements, and the center launch is part of the campaign's broader effort to end cancer through translational discoveries and enhanced patient outcomes. Two findings from a recent study map how scorpions reinforce their weapons with metals. Researchers analyzed 18 scorpion species and found zinc, manganese, and iron deposited in their stingers and pincers to strengthen and sharpen these tools. The study highlights an evolutionary trade-off. Species with more zinc in their tails or pincers tend to have less in the other, suggesting different species optimize different weapon components. Metal enrichment patterns are linked to hunting strategies, whether a species relies on powerful claws or venomous stings, and may reflect broader arthropod evolutionary trends. The research, published in the Journal of the Royal Society Interface, establishes a methodological blueprint for studying metal enrichment across life's diversity and how biology engineers microscopic weapons. On April 28, 2026, the publication date, the article notes a framework for studying metal uptake in scorpions and beyond. Experts say understanding scorpion biology and metal-based weaponry sheds light on their evolutionary success and could reduce fear by underscoring ecological significance. The study carefully accounted for evolutionary lineage to ensure patterns are not simply due to relatedness, strengthening conclusions about metal distribution across diverse scorpion families. This is the first statistical analysis of metal uptake in scorpions that accounts for phylogenetic relationships, suggesting metal enrichment is widespread across roughly 3,000 species. Lead author Sam Campbell notes that metal enrichment has diversified as scorpions evolved changes in how they use claws and stingers. The research used advanced microanalytic techniques, including electron microscopy and X-ray analysis, to locate metal deposition within the weapons. Iron was found more in long, slender pincers than in thick, crushing ones, implying iron adds structural integrity to hold prey for venom action, linking hunting behavior to metal composition. Unexpectedly, species with stronger crushing pincers tended to have less zinc, suggesting zinc enhances wear resistance and hardness where leverage is weaker, while another point links zinc with longer, thinner claws used for venom delivery. A surprising finding in bone metastases shows macrophages with active estrogen receptor alpha signaling, a pattern not seen in normal bone or primary tumors of other tissues. This alpha activity in macrophages was detected in human samples from breast, lung, and kidney cancers, including male patients, pointing to a shared immunosuppressive niche around bone metastases. Baylor College of Medicine researchers developed CEMENT, Sortus A-based microenvironment niche tagging, to map the cellular makeup of metastatic niches across bone, lung, liver, and brain. The method labels normal cells in direct contact with disseminating cancer cells, revealing a consistent feature: abundant macrophages and scarce T cells in these metastatic environments. Across models, the niches show high macrophage presence and reduced T cell activity, indicating macrophages play a central role in local immunosuppression. Pharmacological degradation of estrogen receptors with fulvestrant enables T cells to infiltrate bone metastases and kill tumor cells in both mouse models and human samples, suggesting potential combination therapies. In human tissues and animal studies, blocking alpha signaling facilitates T cell entry and tumor cell killing in bone metastases, pointing toward strategies that pair alpha targeting with immunotherapy. The study, published in Cell as Unbiased Niche Labeling Maps, Immune-Excluded Niche in Bone Metastasis, DOI 10.1016/J.Cell.2026.04.009, highlights a conserved mechanism across cancer types and genders and proposes that targeting alpha signaling in macrophages could enhance immune clearance of metastatic tumors. A new neural network named NEO is trained on matched image pairs from Subaru and the Hubble Space Telescope to reconstruct fine details lost to atmospheric distortion by comparing ground-based and space-based views of the same targets. UC Santa Cruz researchers developed NEO to remove atmospheric blur from ground-based observations and enhance image resolution for the Vera C. Rubin Observatory in Chile. NEO aims to bring Rubin Observatory images closer to space telescope quality by removing atmospheric blur from ground-based data. The model runs on NVIDIA GPU-powered supercomputers and builds on prior web data processing improvements, enabling faster, deeper analysis without replacing astronomers and helping with quicker discovery and pattern detection. Rubin Observatory began science operations in year 2025 and will generate about 20 terabytes of data per night as part of the legacy survey of space and time, creating a substantial data handling challenge that NEO addresses. AI image processing drastically speeds up JWST data analysis, reducing timelines from years to days or less and accelerating potential discoveries. Previous machine learning tools for JWST sped up data processing, enabling earlier identification of mature galaxies. Rubin Observatory, designed to survey the sky every 3 nights, benefits from enhanced image quality through AI to maximize scientific returns on its large telescope investment. The NEO paper detailing its application to Rubin data has been accepted for publication in the Astrophysical Journal, underscoring its peer-reviewed status and potential impact. NEO runs on GPU-accelerated systems and classifies image pixels into objects like stars, disk galaxies, or empty sky, illustrating that AI is intended to augment rather than replace astronomers by handling vast data, flagging unusual objects, and revealing subtle patterns. In testing, NEO improved measurements of galaxy shapes and structures by factors of 2 to 10 compared with standard techniques, potentially allowing Rubin to extract more scientific value without extra hardware. NEO is a conditional GAN with 2 networks, one to generate improved images and another to assess their quality. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.